let's talk about what determines your traits. So when you watch the Introduction to the Basics video, I hope that this is kind of the big picture of what you got. We have DNA, this is your genes, and the genes that you have instructions for how to make all the proteins that make up your cells and do all the interesting work in your cells. Sort of flipping back a little bit to when we were going over nutrition, proteins do almost everything inside of your cells. And so if you have a protein that can do something, that means that that cell can do the thing. Maybe even that organism can do the thing, whether it's digest lactose or process phenylalanine correctly or contract in response to a certain signal or. So our genes contain the instructions for making proteins. The proteins are what are actually doing the things, controlling our functions, giving us abilities or not, as the case may be, and therefore that results in the traits that we see. To make it maybe a little bit more concrete, someone with darker skin has enzymes that are capable of making a specific pigment. Someone with lighter skin has fewer proteins that are capable of making that pigment. Or, in the case of someone with albinism, maybe they have no proteins that are capable of making that particular pigment. So depending on what abilities specific proteins have, that actually shows up in our traits as a human being. Another thing that's interesting about this, specifically when you talk about multicellular eukaryotic organisms, and we'll talk specifically about humans, because that's honestly what we tend to care about the most, is that it's not quite that simple. In fact, as these videos are going to go through, it's not quite that simple at all. We don't just have one gene, one DNA per trait. Generally speaking, we've got two. This picture is a karyotype, that is a snapshot of condensed chromosomes from a cell. This is most frequently done on some sort of fetal or placental tissue in order to check and make sure that the growing fetus has the appropriate number of chromosomes, but you can honestly do this with just about any cell. Sort of above and to the right, we have the snapshot of the dyed chromosomes. This little insert box is all of these rearranged in a fashion that's a little bit clearer, a little bit easier to see. So chromosome 1, for example, is this slightly dull yellow color. So we take this snapshot of chromosome 1 and this snapshot of the other copy of chromosome 1, and we line them up next to each other. We can do the same thing for these two copies of chromosome 2, so on and so forth, get it all lined up, and be able to visually confirm very nicely, yes, this individual has two copies of all 23 chromosomes, and while they may be bent differently, they're the same length, so that implies that they're not missing any chunks, and none of them has any additional chunks that might be bad. And also in this particular individual, we've got two X chromosomes, there's the other one, which means that this individual is chromosomally female, XX, and therefore does indeed have two copies of every single gene. This gets really interesting because it means that there's a backup for everything. There's not just one version of each gene, but two, one from mom, one from dad. Now with this karyotype, it's not possible to really look at this and say, oh, this one is from dad versus that one is from mom, but we do know that we've got the two of them. This is useful in multiple ways. By being able to make the two versions, there's some situations where that means This actually gives an individual more possible traits. It maybe gives them more abilities. And it means that there's a backup. If one of these has been damaged over time by mutation, which we'll talk about later, there's another copy that can still do the thing. So if there's a gene on chromosome 1 that, we'll use the metaphor of it's got the instructions for how to make a bookcase. Now obviously there aren't little bookcases in your cells, but there's a lot of metaphors we have to use in cell biology to keep things comprehensible, reasonable. So bear with me. Maybe mom's copy has instructions for how to make a tall, skinny bookcase, and dad's copy has instructions for how to make sort of a squarish bookcase. An individual with those two versions can actually make two different sorts of bookcases. But what if the bookcase gene from one parent is defective? Well, they've still got the other copy. They still have instructions on how to make a bookcase, and so their cells will still be able to make a bookcase, even if they can only make the one kind. A normal human reproductive life cycle, I sort of mentioned, you're getting one copy from mom, one from dad. Your chromosomally normal, average, however you want to put it, adult human is what's called diploid. Di for 2, diploidy referring to the number of chromosomes. This means you've got 2 copies of each of those 23 chromosomes, or 46 total. In a very few specialized cells in that person's body, in their gamete-producing cells, egg-producing cells, or sperm-producing cells, those are going to undergo a process we'll talk about later called meiosis. The purpose of meiosis is to cut the number of chromosomes in half in order to make, well, actually, let me use a different color here, in order to make eggs and sperm, which are haploid, one ploid, and these each have 23 chromosomes. Why do we have to do that? We do that because the next event is fertilization, where the 23 and the 23 add together to give us the zygote, which is again diploid, 46 chromosomes. But since half are from mom and half are from dad, this is a novel. This is a whole new assortment of those particular genes. The combination that's present in the child isn't the same as either of the parents. This child will grow, develop, and if they end up deciding to reproduce, you're going to get this whole cycle repeating. We'll talk about this a little bit when we talk about evolution. Sexual reproduction is kind of weirdly complicated. You need to find another individual of the opposite mating type and both be producing gametes and have them successfully fertilize before you can make another one. As opposed to, say, bacteria or yeast, where you just split in half, you make 2 complete perfect copies, ta-da, we're done. Why do this? Well, this shuffling, the fact that the parents only contribute a random half each to the child. And so not only is the child very genetically distinct from them in terms of what combination of genes it has, the child is genetically distinct from the other children this couple might produce. And so this provides a lot of fodder for evolution, a lot of diversity within the human species, which can potentially allow us to maybe survive particular famines, natural disasters, diseases better. It's complicated, but it's useful. So what determines your traits? Again, let's wrap this up a little bit. You have 2 copies of every chromosome. The exception to this is if you're chromosomally male. For the 23rd chromosome, people who are chromosomally male will have one X chromosome and one Y chromosome. So they only have one copy of all the genes on those chromosomes. This is one of the reasons that people who are chromosomally male tend to be more susceptible to a lot of genetic diseases that are carried specifically on the X chromosome. Since you've got 2 copies of every gene, one gene is one blueprint. you've got, for the vast majority, if you're XY, or for all of them, if you're XX, you've got a backup. You have a second copy in case of emergency, or just adding that more diversity. And it is always going to be, for those genes where you've got two copies, one from mom, one from dad. This gets really important when we talk about inheritance and Mendelian genetics.